Hello and welcome to my YouTube channel, IB Bio Notes. I will be helping you guys get sevens on your next bio test. So we'll start off with topic 1.1, which is about the cell theory. So Robert Hooke was the first to observe cells using a light microscope in 1665. And after the discovery of the electron microscope, scientists created the cell theory in order to explain properties that were not known before. So there are three parts to the cell theory. The first part is that all living organisms are composed of one or more cells. The second part is that cells are the smallest unit of life. And the third one is that cells always come from pre-existing cells. And you will need to know these three, so write them down. So for the second part, we have atypical cells, which are cells that contain an unusual characteristic from typical cell characteristics. So the first example would be here in the top right, a striated muscle cell, and they're composed of repeated units that are called sarcomeres, and they're atypical since they're much bigger than an average cell, and they also have multiple nuclei. The second one we see here is the giant algae, and this is composed of a huge single cell, and it's made of three parts: the roots, the stalk, and the top umbrella. And the reason it is atypical is because it is much larger than average cell, and it is not simple in structure. The third example we have is the ascipitae fungal hypha, and it is composed of long threads with many nuclei and no cell wall, which results in a shared cytoplasm and a bigger than average cell, as we can see here. And this is also why these are all the characteristics that make it atypical. Now, the units of measurement we'll need to know. So a typical animal cell ranges from 10 to 20 micrometers. So here, these are the units of measurement we'll have to memorize. So 1000 nanometers are equal to 1 micrometer. Nanometers are the smallest unit of measurement. 1000 micrometers are equal to 1 millimeter, and so on. So magnification, we'll have to know how to calculate that, is the size of the drawing over the actual given size. So, for example, in this picture, when it gives us the key here, we're going to measure this using a ruler and say this is 2 centimeters. So we're going to do the size of the drawing over the actual size. So we'd have 2 centimeters over 100 micrometers, because this is given. So 2 centimeters, we convert that to millimeters, that would be 20 millimeters. We convert that further to micrometers. So it'd be 20,000 micrometers over 100 micrometers. And we'd get a total of 200 times magnification. Yeah. So for the next part, we have unicellular organisms. So these are living organisms that are composed of only one cell, such as these given examples. So paramecium is an example you'll most likely have to know, and it is right here. It's a single cell organism. They're heterotrophs, and they move with cilia, which are these small hair-like structures that help in their transportation. And you'll definitely need to write these down. They're really important. The 7 functions of life that all cells need to undergo are, firstly, is metabolism, which are all the life-supporting chemical reactions that are inside the cells or any living organism. Second one is growth, which is the increase in size or shape of a cell over time. Third one is response to external environment or stimuli. And homeostasis as well. It is the maintaining of the internal conditions inside of a cell. The fifth one is nutrition, which is the intake of nutrients such as making organic molecules in photosynthesis or absorbing it for animals and fungi. The sixth one will be reproduction, which is the production of offspring, either sexually or asexually, to pass on their genes onto their offsprings. And in unicellular organisms, this is always asexual reproduction. It is called binary fission. And lastly, we have excretion, which is the removal of waste products of the metabolism through the cell. So the surface area to volume ratio is why cells usually divide. So we know that cells' volumes increase much faster than their surface area. So the cells obtain a smaller surface area to volume ratio as they grow, and they aren't able to import molecules and expel their waste efficiently. So in order to survive, they have to split into two daughter cells so that a larger surface area to volume ratio can be restored and they can excrete their waste and obtain nutrients more efficiently. So this is the journey to multicellular organisms. So first of all, cells always started off as unicellular, and until these organisms grew larger, because they were no longer limited by the size of one cell. And the cells in these organisms were able to specialize through differentiation, which is a process in which an unspecialized stem cell develops into new cells with more distinct structure and function. And these multicellular organisms displayed emergent properties, which basically means that the whole organism can do more than just the addition of its separate parts because of its interaction between these different parts. You'll have to know these definitions: differentiation and emergent properties. And so you also have to know the genome, which simply refers to the complete set of genes or genetic material present in a cell or of an organism. So the human genome consists of around 21,000 genes, and these genes are existent in all of our cells. However, only some of them are expressed in each cell, which is what causes each cell to be unique and differentiate and specialize. So we have stem cells, which we established before, are undifferentiated or unspecialized cells, and they can arise, and create different types of cells. So we have four types of stem cells. The most unspecialized type is totipotent stem cells, which are the eight cells of the morula, which are the cells following fertilization of an egg, and they can differentiate into any cell and give rise into and create an entire organism. The second type is a pluripotent stem cell, which can differentiate into any cell, but they can't create a whole organism. An example of that would be embryonic stem cells. The third type would be multipotent stem cells, so for example from an umbilical cord, and they can differentiate into a few types of blood cells. And the most specialized type of stem cell is a unipotent stem cell, and they can only differentiate into their associated type. So for example, a liver or heart stem cell can only differentiate into liver or heart cells. So diseases involving stem cells are Stargardt's disease and leukemia. We'll have to know both of them. So Stargardt's disease is a disease of the eye, and it is a form of juvenile macular degeneration. And it is treated by injecting retinal stem cells that are derived from human embryos, and they're injected into the patient's retina, which attach to the retina and become functional. And similarly, leukemia is a type of blood or bone marrow cancer, and is caused by high amounts of abnormal white blood cells. And it's a complicated process in treating it. But first, hematopoietic stem cells are extracted from bone marrow or an umbilical cord, and then the patient receives chemotherapy to kill off the diseased white blood cells. And then these hematopoietic stem cells that are extracted earlier are then transplanted back in to differentiate into healthy white blood cells. So the ethical issues and concerns surrounding stem cells. So some methods of obtaining stem cells actually involve destroying a specifically created embryo, and so these ethical concerns arise. So how are embryos specifically created? DNA is removed from a donor's egg cell, and host DNA is added from somatic cells. And the egg cell then develops into a blastocyte and provides embryo stem cells. So the question here is it fair to terminate a potential life to save a person? So there are a few reasons to support it. So one would be to eliminate serious diseases in the human population. It could help in organ transplant without the loss of a life or pressure on the body. And they are harvested early on, so embryos can't really feel pain. However, embryos are also considered living beings, and creating life to terminate it goes against moral, religious, and cultural guidelines. So that is it for topic 1.1. Thank you for watching.